Hello everyone, Science Spot is a podcast that shines a spotlight on the latest scientific technology from Japan. Your host is Ren from Science Talk. Today I'm going to talk about the discovery regarding the mechanism of aging cells. This research was announced by a collaborative group from the Kyoto University, University of Tokyo, and RIKEN. First, what exactly is cellular senescence? It's a phenomenon where our body's cells, when exposed to various stresses such as oncogene activation, DNA damage, or replication stress, irreversibly stop dividing. Originally, this cellular senescence serves a crucial protective function. By preventing damaged cells from proliferating, it helps suppress cancer. However, it has also been found to be deeply involved in many age-related diseases, including lifestyle-related disease and neurodegenerative conditions. Furthermore, a very characteristic feature of senescent cells is something called SASP, which stands for Senescence-Associated Secretory Phenotype. SASP is a general term for the phenomenon where senescent cells secrete various bioactive substances that can cause inflammation or even promote senescence in surrounding normal cells. You can think of it like a single rotten apple in the barrel, which simply by being there starts to spoil the healthy apples around it. One previous studies using cultured cells suggested that the SASP could affect surrounding cells. The precise mechanism of how a tiny number of senescent cells in our bodies contribute to widespread aging throughout the entire organism has remained a significant mystery until now. It has been challenging to accurately identify and track these senescent cells within the living body. The research group developed a groundbreaking new mouse model that allows them to directly observe and track senescent cells within the living body. Specifically, they created genetically modified mice that could artificially and constitutively activate two key signaling pathways, the ERK pathway and the P38 MAPK pathway, which are known to be involved in the cellular senescence. By administering a specific drug called doxycycline, and these pathways are activated, thereby inducing cellular senescence in vivo. What they found surprisingly was when they directly induced the senescence in cells using the doxycycline, and not only did these cells become senescent, but even the healthy cells surrounding them also started to age, as if caught in the chain reaction. This is a clear evidence that senescence propagates, meaning secondary cellular senescence occurs. To observe this propagation in more detail, the research team developed a unique two-color labeling system. Primary senescent cells were labeled with red fluorescence, and secondary senescent cells were labeled with green fluorescence. This allowed them to accurately visualize and analyze how cellular senescence spread and affects cells within the living body at a single cell level. Another crucial finding from this model was the manner of cellular senescence varied significantly depending on the tissue type, the factors that induce it. For instance, the cellular senescence response was very different in various cell types, such as a liver, large intestine, and mouse embryonic fibroblasts. Even more surprisingly, within the same type of cell, like liver cells, the senescent state was not uniform, but it was suggested to consist of multiple cell populations with diverse gene expression patterns. This suggests that just like fire splats differently in a wooden house compared to reinforced concrete building, cellular senescence is not a uniform process, but rather exhibits diverse characteristics. The liver has a precise structure called zonation, which consists of three functionally distinct regions. They found that inducing cell senescence, especially through ERK pathway activation, disrupted this intricate zonal structure of the liver, potentially leading to a decline in liver function. This is an important insight, confirming that the changes at the cellular level can impact the function of an entire organ and are closely related to individual aging. So how does this secondary cellular senescence, this chain reaction where surrounding cells age, actually occurs at the molecular level? The research group found a crucial clue to this secret conversation. By thoroughly analyzing the comprehensive gene expression data of secondary senescent cells, they discovered that information transmission pathway called IL-1 signaling plays a vital role in inducing the secondary senescent cells. Further investigation revealed that the primary source of this IL-1 signal, particularly in the liver, is a type of immune cells called macrophages. Macrophages are known as eating cells because they clear foreign invaders and cellular debris from the body, but they also secrete an inflammatory cytokine called IL-1 beta, which acts as a communication substance between cells. In this research, they demonstrated that when they used a drug to selectively deplete macrophages throughout the body, or a drug to inhibit the receptor that receives IL-1 beta, the expression of the marker for secondary cellular senescence was suppressed. This strongly suggested that IL-1 beta secreted by macrophages acts like a signal interacting surrounding cells to get old, thereby triggering the chain reaction of aging. It's as if at a fire scene, the firefighter who should be leading the extinguishing efforts were actually giving the command to start a fire, a truly astonishing discovery. This research represents a groundbreaking achievement, as it is the first time that the behavior primarily and secondary senescent cells in the living body and their impact on the surrounding cells has been clarified with a single cell resolution. The new mouse model developed in this study expected to significantly accelerate future aging research and in turn contribute a fundamental basis for the development of completely new treatment strategies for various age-related diseases. I think most people don't want to age, and I certainly don't either. That's why I'm really looking forward to advancement in its research like the ones we discussed today, as I believe they'll give us a much clearer picture. While there are many reported cases in aging research, human studies are still very much in their early stages. So if there are any updates, I want to cover them on this podcast again. That's all for today's Science Spot. This podcast is broadcast daily on weekday morning in both Japanese and English. I hope today's discovery has changed how you view your thought, and I'd love for you to listen to the podcast and post your notes and thoughts with the hashtag ScienceSpot. See you next time.